Vagina is a fibromuscular tube partially covered by a membrane called the hymen. Structure: vagina consists of 3 layers: mucosa, musculosa, and adventitia. Mucosa is transversely folded and consists of epithelium, stratified squamous non-keratinized epithelium rich in glycogen, which when released the cells appear vacuolated. Lactic acid is not released by the vaginal mucosa, but the bacteria flora of the vagina takes the glycogen to manufacture lactic acid. The epithelium undergoes cyclic changes. Before puberty and after menopause, the epithelium is thin, but during the reproductive years, the epithelium thickens due to increased mitotic activity as a result of estrogens. The superficial layers increase in number and size due to accumulation of glycogen and lipid within the cytoplasm. Corium is formed of dense connective tissue. There are no glands in the vagina, but the vagina is kept moist by secretions from the cervix. But the lamina propria contains capillary plexus, which is the source of fluid exudate from the vagina during sexual stimulation. Musculosa of the vagina is formed of interlacing fibers arranged as inner circular and outer longitudinal layers. Then adventitia is composed of fibrocollagenous tissue containing thick elastic fibers, large blood vessels, and nerves. The extensive elastic fibers in the wall of the vagina is responsible for elasticity that permits the great temporary distension required during parturition. Comparison between esophagus and vagina. Esophagus is a long, simple passage of the alimentary tract, while vagina is a short, female excretory passage. Esophagus has a narrow lumen and longitudinal mucosal folds, while vagina has a wide lumen and transverse mucosal folds, not well apparent. Structure: esophagus consists of mucosa formed of epithelium, stratified squamous, partially keratinized in certain animals. Corium poor in elastic fibers, and there is the presence of muscularis mucosa. Submucosa consists of loose connective tissue and may contain mucous glands in certain animals like dogs and in humans. Musculosa is striated in the upper third, mixed in the middle third, and smooth in the lower third. It is well differentiated into inner circular and outer longitudinal layers, then adventitia of loose connective tissue. Vagina. Its mucosa consists of stratified squamous epithelium rich in glycogen and may appear vacuolated. Corium is rich in elastic fibers. There is no muscularis mucosa. There is also no submucosa. Musculosa is smooth, not well differentiated, and it is formed of inner circular and outer longitudinal layers. Then adventitia consists of thin connective tissue rich in elastic fibers and blood vessels. Fertilization. Ovum, oocyte,
undergoes maturational changes prior to fertilization in the ampulla of the uterine tube. Second maturation division. Only a single spermatozoon penetrates the ovum. This is rapidly followed by reduction of permeability of the ovum's zona pellucida, thus destroy sperm receptors in zona pellucida, preventing entry by other sperms. This is a chromosome reaction. This results in the entry of sperm nucleus and fertilization. Zygote two-cell stage is formed 30 hours after fertilization. Then a morula formed 72 hours after fertilization, which is 8-cell stage, covered by zona pellucida, which is formed by successive mitosis of the zygote, fertilized ovum. The cells resulting from division of the zygote are called blastomeres. There is gradual accumulation of liquid between the cells of the morula, resulting in formation of blastocysts. Human blastocyst has an inner cell mass that will form the embryo proper, and a peripheral single layer of cells, trophoblast, that will form the fetal part of the placenta, chorionic plate. Blastocyst breaks zona pellucida and remains free in the uterine cavity, and will implant in the endometrium during the secretory phase, 6 to 7 days after fertilization. This is a diagram showing the process of fertilization of the ovum and its implantation in the endometrium of the secretory phase.
Placenta formation. After fertilization of the ovum in the fallopian tube, the fertilized ovum changes to morula and descends to be embedded in the uterus. The placenta is now formed of two parts, a fetal part and a maternal part. The endometrium is called the decidua and is divided into decidua basalis between the embryo and the myometrium at the implantation site and forms the maternal part of the placenta. Decidua capsularis covering the embryo between it and the lumen of the uterus. Then decidua parietalis, which is the rest of the decidua. The fetal part of the placenta is formed of finger-like projections, chorionic villi, arise, branch, anastomose, and are embedded in the endometrium. Each chorionic villus consists of an epithelial covering made of two layers, inner cytotrophoblast or Langerhans layer formed of cubical epithelium, rich in glycogen, and outer syncytial trophoblast. Then the villus has a core of connective tissue rich in mesenchymal cells and containing the fetal blood. After 4 to 6 months of pregnancy, the immature placenta becomes mature and the layer of cytotrophoblast disappears, thus forming the terminal villi, each consisting of a core of connective tissue rich in mesenchymal cells and containing the fetal blood vessels, and is covered by a layer of syncytial trophoblast only. Placental barrier is the barrier between maternal and fetal blood. It consists exclusively of fetal tissue. Syncytial trophoblast, cytotrophoblast, plasmid membrane of the trophoblast, connective tissue, plasmid membrane of the fetal capillaries, and endothelial lining the fetal capillaries. Functions of placenta. It supplies the fetus with nourishment, oxygen, and removes residual products of metabolism from fetal blood to maternal blood. Secretion of hormones as human chorionic gonadotrophins, which its presence in urine is used in pregnancy tests to diagnose pregnancy. Placenta is a site for storage of carbohydrates, proteins, calcium, and iron to be used by the fetus. Also, it forms the placental barrier, which prevents harmful materials from reaching the fetus from the mother. Umbilical cord runs from the fetal umbilicus to the placenta. It contains two arteries which carry deoxygenated blood from the fetus to the placenta, and a vein which carries oxygenated blood to the fetus after losing carbon dioxide and taking oxygen from the mother. The vessels are embedded in mucous connective tissue called Wharton's jelly. Umbilical cord is covered by a amniotic sac of a single epithelial layer early in pregnancy, which becomes stratified late in pregnancy. Mammary glands are identical in both sexes prior to puberty. In males, they remain rudimentary, but in females, they change in size, shape, and structure with age and functional status. Shape. Mammary glands are conical in shape, having a cylinder projection, the nipple, surrounded by a circular pigmented skin area called areola. Structure. Mammary gland is a compound tubuloalveolar gland formed of lobes and lobules. The lobules are separated by dense and fatty connective tissue. The parenchyma of the mammary gland is formed of ducts and alveoli embedded in connective tissue. But this varies according to the state of mammary gland. Resting mammary gland has collapsed alveoli represented by solid cores of cells, few ducts embedded in abundance of fatty connective tissue, while lactating mammary gland has wide alveoli lined by tall columnar or low cuboidal cells depending on the state of activity. The alveoli are distended with milk. There is abundance of ducts and little amount of connective tissue. Myoepithelial cells are found around the alveoli. After menopause, there is involution of the mammary glands, structural and functional atrophy of both secretory portions and ducts. Comparison between thyroid gland and lactating mammary gland. Thyroid gland is an endocrine gland, has no ducts. It is covered with a connective tissue capsule and has incomplete septa and fine reticular fibers. Its parenchyma is made of follicles and interfollicular tissue. The follicles are lined by cubical cells and C cells or parafollicular cells. The interfollicular tissue consists of reticular connective tissue rich in fenestrated blood capillaries and various interfollicular cells. Thyroid gland is a merocrine gland secreting thyroxine by follicular cells and thyrocalcitonin by parafollicular cells or C cells. The colloid secretion in the follicles is homogeneous acidophilic mass. Lactating mammary gland is an exocrine gland with ducts. It is covered with skin and adipose connective tissue. Lactating mammary gland is a compound tubulo-alveolar gland. It is formed of lobes and lobules, separated by interlobular connective tissue containing blood capillaries and different ducts, and various connective tissue cells. The alveoli of lactating mammary gland are lined by either tall columnar or low cuboidal cells depending on the state of activity. Lactating mammary gland is an apocrine gland secreting milk. Milk present in the alveoli is different from colloid in the follicles of the thyroid gland. It is not homogeneous but vacuolated due to dissolved fat globules.